Welcome back to part 2 of our hormones topic. In this part, we looked the first part at how these hormones are secreted and where from, and then how they get around the body in a little way, and how they're then controlled by negative feedback. In this part, we'll look more at what happens when they get to their target cells. Now, a lot of that depends on whether they're hydrophobic or hydrophilic. That's going to determine where their receptors are, whether they can get through the membrane or whether they have to bind extracellularly. So we will meet our old friend, the G protein. Those classes, those three classes, vary in terms of hydrophobic and hydrophilic. So we'll look at those classes, we'll look at the signaling mechanisms, and then we'll very briefly really look at how the effects of hormones can be up and down regulated, can be amplified, and which brings us back really to understanding why negative feedback is so important. Enjoy. Okay, so our three classes of hormones then. We'll start with the biggest. Protein hormones, sometimes called peptide hormones, polypeptide hormones. This is the biggest group. So it includes all of the hormones synthesized in the hypothalamus and in the anterior pituitary. So that's a big group of hormones. It also includes insulin and glucagon. These are both protein hormones. And it includes parathyroid hormone and erythropoietin. Steroid hormones, the ones that we're really going to focus on, are cortisol and aldosterone. Cortisol being our glucocorticoid, aldosterone the mineralocorticoid. Together they're called corticosteroids because they come from the adrenal cortex. Steroid hormones includes a few others, things like vitamin D from the kidneys, DHEA, you really don't want to know what that stands for, androstenedione. These are the precursors of the male and female sex hormones, and they come from the gonads. Our other group are amine hormones, also have multiple names, I'm afraid, might be known as amino acid hormones or derivatives of tyrosine. Tyrosine is one of the amino acids, and it forms a constituent part of the amine hormones. This group includes thyroid hormones, T3 and T4. So think about our protein hormones. Although TRH and TSH are protein hormones, they stimulate the secretion of an amine hormone. So not all the hormones within an axis have to be of the same type. So we have T3 and T4 are amine hormones. They come obviously from the thyroid gland. It also includes catecholamines. Catecholamines is a sort of umbrella term, or a small umbrella anyway, for adrenaline and noradrenaline. These come in terms of hormones from the adrenal medulla. So protein hormones are a big group. Most hormones, as we said, are peptide or aka protein hormones. They range in size from a couple of amino acids up to nearly 200 amino acids, all in a nice long folded polypeptide chain. They are water soluble. In other words, they are hydrophilic. They are, clues in the name, proteins. So they're synthesized by protein synthesis. So they're synthesized in the ribosomes in those specific endocrine cells. When they're first synthesized, they are large, inactive pre-pro hormones. And this is where our cleaving of protein starts to come in. So you can see there we've got pre-pro insulin. Looks a bit like a paperclip. We've got a little blue signal sequence on the end. This is an inactive pre-pro hormone. That is then cleaved by enzymes, not by a little chopper, but a chopper is more fun. These are then cleaved to become prohormones, and this happens in the endoplasmic reticulum. They're still not active though. They're transferred into the Golgi apparatus from the endoplasmic reticulum, come out of the Golgi apparatus, and then they're stored in vesicles in the cytoplasm, waiting for a signal that will trigger their exocytosis. But before they're exocytosed within the vesicles, they're cleaved again, and they're cleaved again to become the active hormone. And you can see what's happened here. First of all, we lost the signal sequence. Now we've lost, in terms of insulin, our chain C. This has left us with chain B and chain A, very simple structure, and this is our active insulin. They're then exocytosed into the circulation, off they go, and go around looking for specific receptors to bind to on target cells. They will only bind to cells that have those receptors. Steroid hormones, as we said, we have our corticosteroids. These are synthesized and secreted by the adrenal cortex, the outer bit of the adrenal gland. So we have our mineralocorticoid, our aldosterone, we have our glucocorticoid, our cortisol, and we also produce some androgens. The ovaries and placenta and the testes, they also produce steroid hormones. So our adrenocortical hormones can also be called corticosteroids. So as we said, we have our mineralocorticoid, which is aldosterone, our glucocorticoids. It's not only cortisol, there's also corticosterone. Corticosterone can be converted then to aldosterone. And we have our androgens. We've got DHEA and androstenedione. These are the precursors of the sex hormones. And that's probably the last we're going to mention of those. Steroid hormones are synthesized as needed, and they're synthesized from vacuoles of cholesterol because they are cholesterol based. And this happens within the endocrine cell. Of course, they're synthesized as needed because they're made of cholesterol, and we've already said they are actually hydrophobic, lipophilic. So if we made them in advance, there's a good chance that they might start to move through plasma membranes and we'd lose them. So they're synthesized as needed. So because they're synthesized from cholesterol, which is one of our lipids, they are lipid soluble. They are hydrophobic or lipophilic. Because of this, they can diffuse through the endocrine cell's plasma membrane out into the interstitial fluid. They can then diffuse into a target cell, so therefore they bind intracellularly. Amine hormones, as we said, we've got our thyroid hormones, T4 and T3. Now remember that the 4 and the 3 relates to the number of iodines. Iodine carries a negative charge. That makes our T4 and T3 slightly negatively charged. And if you remember, nothing with a charge can diffuse through the membrane. So although in this case thyroid hormones are technically lipid soluble, they can't diffuse through the plasma membrane because of the charge on the iodines. Therefore, they do bind intracellularly, but they can't get through the plasma membrane, so they have to go through these special transporters, iodothyronine transporters, to get into cells. Our amine hormones also includes our catecholamines. So we have noradrenaline. If you're American, you might call it norepinephrine, or you might see that in text. And we have adrenaline, epinephrine. We will use noradrenaline and adrenaline. So these come from the adrenal medulla. So whereas our corticosteroids came from the cortex, the outer part of the adrenal gland, these come from the inside, from specialist cells known as chromaffin cells. They are water soluble, so just like our protein hormones, they have to bind on the outside of cells to receptors, and these are generally termed adrenoreceptors. One specific one you've probably heard of are the beta-1 receptors or adrenoreceptors. In terms of the adrenal medulla, it tends to produce more adrenaline than noradrenaline from these chromaffin cells. And this is quite logical, really, because we also get noradrenaline exocytosed as a neurotransmitter from sympathetic neurons. And if the sympathetic nervous system is activated, that can result in a lot of noradrenaline floating around. But both noradrenaline and adrenaline generally will bind to the same adrenoreceptors. Strangely enough, their exocytosis is stimulated by ACTH. If you remember ACTH from the anterior pituitary, stimulates the release of cortisol from the adrenal cortex, but it also stimulates the release of these catecholamines from the adrenal medulla. Other stimulation of our noradrenaline and adrenaline will come from efferent sympathetic neurons. So sympathetic neurons run down to the adrenal gland, send an action potential to stimulate the secretion of noradrenaline and adrenaline. So you can see these catecholamines work very closely with the sympathetic nervous system. Their role really is generally upregulation. And if you remember, the sympathetic nervous system is all about fight or flight. This is our fight or flight response. So everything tends to get upregulated. If you're going to run away or if you're going to fight, you need your heart rate up. You need your respiratory rate up. You need your heart to pump harder. Everything tends to get upregulated in this. Their general role the rest of the time is really in maintaining homeostasis. The autonomic nervous system controls a lot of organ function, of visceral functions, without us knowing that they're doing it. So that was our classes of hormones. Because of this difference between them in terms of whether they're water soluble or lipid soluble, that is going to impact on how they signal. So receptors for water-soluble hormones are found on the surface of the target cell. They can't get in, they have to bind outside. These are transmembrane proteins. But then on the inside, we need something to take that signal and carry it to the relevant organelles or to the nucleus. So we need a second messenger system. And this brings us back, if you remember our old friend the G protein, he's one of the initiators of whole cascades of signaling that happens within cells when a receptor is bound. So many of these processes, these intracellular signaling processes, signal transduction, involve a G protein. There are others, and we will talk about one. Receptors for the lipid-soluble hormones, of course, the lipid-soluble hormones can go straight through the plasma membrane, so their receptors are going to be inside the cell, either in the nucleus or in the cytoplasm. One thing to remember, though, is that the receptors are specific for one or sometimes two, think of noradrenaline and adrenaline, ligands, in this case hormones. So one hormone will bind to one receptor, that old lock and key concept. And this helps to identify the target cells for individual hormones. Remember, these are endocrine hormones. They go whizzing around the circulation. They don't know where to bind. What tells them where to bind is the presence of receptors on target cells. If they don't have those specific receptors, the hormone will have no action on that cell. If they do have them, target cells generally, the number will vary quite widely, but it is very high. So you might have 2,000 to 100,000 specific receptors for one hormone on or in one cell. These can be upregulated or downregulated depending on other factors. So our protein hormones then, and the catecholamines, signal via extracellular receptors. This means they need second messenger systems, so there has to be something on the inside. And here's our G protein. G proteins can do various things. They can activate or inhibit an enzyme called adenylate cyclase. They can activate another enzyme called phospholipase C, or they can activate or inhibit ion channels. Historically, and so far up to this point really, we have always focused on the alpha subunit GA as doing all the work. These subunits were only identified in 1980, and it's recently been shown that the other two subunits, we've always said about beta and gamma being left behind at the bus stop, but these other two subunits together as a dimer, two things stuck together, and that's G beta gamma, can also interact with the same enzymes and channels. And the effects of this are still not terribly clear, and they're still finding out an awful lot about what beta and gamma can do together, while alpha has gone off to have his fun. The one difference is, of course, that the alpha subunit GA has the GTP. He definitely takes the GTP with him. He has the GTP because he needs to hydrolyze that, so he must have GTPase built into him, so that the alpha subunit can return to our beta and gamma subunits to form the inactive whole G protein to be used again. After we've looked at G proteins, and we'll look at G proteins in terms of what they can activate, just to even things out about a bit, we'll look at a process that doesn't involve G proteins. It's a very key and important process in terms of physiology and pathology, and these are tyrosine kinase receptors. So first of all, let's have a look at our G protein. Let's have a bit of revision about activating a G protein. So hopefully this will look familiar. Remember, a hormone binds to the receptor, and our intracellular G protein, remember this is a peripheral protein, binds to the intracellular end of the receptor. So we have hormone bound at the top, G protein at the bottom. Can you remember what happens next? Yep. The alpha subunit, which was carrying GDP, which is useless to it, gets rid of GDP and takes on GTP. And next, the whole G protein moves away from the receptor. If you remember, this is because as long as that, in this case, hormone is bound to the G protein coupled receptor, more G proteins will come up and bind and be activated. So it needs to move out of the way. So the whole G protein moves away from the receptor. After that, our alpha subunit, taking the GTP, separates from our beta and gamma subunits. Beta and gamma stay together. That's one thing that has become clear. They might go off and do things, but they do stay together as a dimer. As we said, as long as the hormone is bound, more G proteins associate and are activated, and they'll go through exactly the same process. Okay, so our alpha subunit, or our beta and gamma together as a dimer, can activate or inhibit various things. This, what they do activate and inhibit, depends on the receptors present on that cell or the type of G protein associated with that receptor. We can call them different things. So GS, the S stands for stimulatory. Don't ask me what the Q stands for, but it's also stimulatory. So GS and GQ are stimulatory. GS is the G protein that stimulates adenylate cyclase. GQ stimulates phospholipase C, which we'll talk about a little bit later. GI are inhibitory. So we have inhibitory G proteins, and each receptor will be associated with a different type of G protein, whether it be stimulatory or inhibitory. They're all activated in the same way, so even if they're inhibitory, we have to activate them first so that they can inhibit. So the type of G protein associated with the receptor depends on the type of receptor that the hormone binds to. And hormones may have multiple different types of receptors. The action of the G protein within the cell is also dependent on the receptor. Therefore, hormones may have different actions in different cells. So if it binds to a receptor on one cell, it might be stimulatory of a certain something. If it binds to a receptor in another cell, a slightly different type of receptor, then it might be inhibitory of something in particular in that cell. So in different cells, G proteins, hormones may have different actions. But even in the same cell, there may be different receptors. So you may have receptors that have different actions. So several molecules of your hormone will bind to different G protein coupled receptors expressed on the plasma membrane, and this might cause a variety of different actions within the cell. Yeah, it all gets very complex. This is why we're not going too deep in. So one of the things that our G proteins can do is activate or inhibit adenylate cyclase. You may see this in text as adenylyl cyclase or adenyl cyclase. We'll use the term adenylate. Adenylate cyclase is an enzyme, and it's an enzyme that generally is membrane bound. So in other words, it's embedded in the plasma membrane. Every cell has quite a few of these adenylate cyclase enzymes. When it's bound and activated by a GS protein, what it does, it takes ATP, it doesn't use it for energy, it converts it to cyclic AMP, so monophosphate, which releases a couple of phosphates. Phosphates we don't care about, they go off and join their mates all floating around the cell and have a bit of a party. Cyclic AMP, on the other hand, or CAMP, or CAMP, has lots of other functions. This is just the start of a signaling cascade. However, if an adenylate cyclase is bound by a GI protein, so this is will be from a different receptor, it blocks adenylate cyclase. So it stops adenylate cyclase taking ATP and producing cyclic AMP. In this way, with all the adenylate cyclases within a cell, if we stimulate more GS, we'll get more cyclic AMP. If we stimulate more GI, we'll get less cyclic AMP. So it's actually quite a good way of regulating the signaling cascades going on with inside a cell, a bit like a seesaw going up and down. So cyclic AMP up a bit, cyclic AMP down a bit. GS, they go up, GI, they go down. So it regulates cyclic AMP levels within the cell. And we'll come back to a lovely example when we're doing cardiovascular. Now, cyclic AMP, as we said, has lots of roles in its own right. This is the beginning of a signaling cascade. One of the things cyclic AMP can now do is activate another enzyme called protein kinase A. The thing with anything with the word kinase in it means that it phosphorylates. Any kinase, kinases, they all phosphorylate. This one is known as protein kinase A, or pKa. So protein kinases, when activated by cyclic AMP, phosphorylate other enzymes. And this can have a stimulatory or an inhibitory effect on those individual enzymes. So you can see the level of complexity we're getting to here. Yeah, we may have some cyclic AMP. Yes, we've activated a protein kinase, gathers up all those free phosphates and chucks them out to a load of enzymes. In some of those enzymes, that might inhibit them. In some of those enzymes, it might stimulate them. But we've got a complete amplification of the effects of one cyclic AMP. The other thing that these phosphates can do, other than stimulating or inhibiting other enzymes, is to sensitize channels. And this is quite a key signaling mechanism, really. So protein kinase A will send out phosphates to channels. When you phosphorylate a channel, it usually sensitizes them. What we mean by that is that that channel will open more readily. Whatever the gating mechanism is, it will need less stimulation to open because it's been phosphorylated. Eventually, cyclic AMP is inactivated. It's degraded by another enzyme, phosphodiesterase. Okay, another thing that our stimulatory G protein can activate, as we said before, this is actually GQ, but let's just stick with GS. GS for stimulatory, GI for inhibitory. The other thing it can do is activate another enzyme. This is also a membrane-bound enzyme, has to be membrane-bound, this one, called phospholipase C. We'll come back to another phospholipase later. Phospholipase C sits in the membrane. It's an enzyme that can break down phospholipids to use them for other things. So, GS comes along, binds to and stimulates our phospholipase C. Abbreviations that, again, you might see in text would be PLC. PLC then converts a specific type of phospholipid. It's a small, about 1–2% of the phospholipids are these particular phospholipids PIP2. Don't worry about the full name. PIP2 is absolutely fine. PIP2 are phospholipids that are found in the plasma membrane. So phospholipase C takes those PIP2 phospholipids and it converts them into two other things. So you get a bit of a bog off here. Buy one, get one free. Because you're actually producing from one activation by our GS, you're actually producing two different things. The first is DAG, diacylglycerol. DAG, to be honest, you can use the term DAG, that's fine. So it produces DAG and it produces something called IP3, inositol triphosphate. Again, the abbreviation is fine, IP3. DAG and IP3 have different actions. So we'll start with DAG. DAG activates protein kinase C. So we've got another protein kinase. There it goes. It activates protein kinase C. Protein kinase C at this point generally is floating around the cytosol near the membrane. Once it's been activated, protein kinase C moves to the membrane, and being a kinase, it then phosphorylates other proteins to activate or inhibit them. So on the one hand, we've got DAG activating a protein kinase, which then phosphorylates other enzymes. So that was DAG. What about IP3? Well, the endoplasmic, or in myocytes, we call it the sarcoplasmic reticulum, always has within it a store of calcium, Ca++. IP3, when it's produced by phospholipase C, then goes off and binds to specific receptors on the endoplasmic or sarcoplasmic reticulum. This opens calcium channels in the membrane of the endoplasmic sarcoplasmic reticulum. So calcium, of course, high concentration in there, then follows its gradient and moves out into the cytosol of the cell. One of the things that calcium can do, and this converts it into a signaling molecule rather than an ion, rather than something that's going to stimulate contraction, it binds to calmodulin. Calmodulin combined with calcium becomes a signaling molecule. So it can go off and it can cause lots of other reactions. Of course, calcium can also cause or increase contraction in myofilaments. So this can affect myocytes, so we might increase the strength of contraction of a myocyte, or it can stimulate exocytosis. Remember, all exocytosis involves the interaction of calcium with the cytoskeleton, with the myofilaments of the cytoskeleton, which helps to push vesicles towards the membrane so that the contents can be exocytosed. So this calcium can combine with calmodulin to become a signaling molecule, or, depending on the cell, it can cause or increase contraction of myofilaments, which may cause contraction, may cause exocytosis. So we got two deals really for the price of one. We got DAG activating protein kinase C, which gave us phosphorylation of enzymes. We get IP3, which releases calcium from the endoplasmic or sarcoplasmic reticulum. Which brings us on to amplification, because the one thing that we've seen here is that all of these signaling cascades, and we've scratched the surface really in terms of these signaling cascades. We said about phosphorylation of other enzymes may activate, may inhibit them. They're going to then interact with other enzymes and do other things. So we've got this massive cascade of effects from one molecule of hormone binding. So in some cases, we could get a single hormone molecule binds to an extracellular receptor. And inside the cell, we can activate 100 G proteins in the time that it is still bound to that receptor before the hormone is inactivated. Each G protein activates one adenylate cyclase if it's stimulatory, or inhibits one if it's not. Each adenylate cyclase molecule, if it's stimulated, produces 1,000 molecules of cyclic AMP. Each cyclic AMP can activate a protein kinase. We now have 1,000 protein kinases activated. Each protein kinase can act on thousands of enzymes. You think about all those phosphates just chucking out. Oh yes, inhibit you, activate you. Massive, massive effects in terms of a tiny, tiny cell. So as an example, one adrenaline molecule, remember adrenaline is one of our catecholamines, generally released as part of the fight or flight response. So this adrenaline molecule can result in the breakdown of millions of glycogen molecules into glucose. So think about it, fight or flight. You might want a bit of glucose flowing around your blood to make lots of ATP because you're going to need lots of ATP. So one molecule of adrenaline can break down millions of glycogen molecules. The other thing to consider is something that we talked about way back, that receptors can be upregulated or downregulated. So this can affect the affinity of receptors, the attractiveness of them. So we can take down some receptors that are not very attractive, have a low affinity, and put up high affinity ones, or vice versa. We can also take down some receptors, so we're expressing on our plasma membrane fewer of them, or we can exocytose more receptors, so we're expressing more of them. That is going to have an impact on the action of our hormone. The more receptors you have, the more impact it's going to have. The more affinity you have, the more impact it's going to have, and vice versa. The other thing to consider is that the same hormone may use different second messenger systems in different cells, and in some cases, even within the same cell. Okay, so moving on to our one example without a G protein. It's quite an important one, tyrosine kinase signaling. We have no G proteins involved, but it's really important in terms of the regulation of blood sugar because you remember insulin came from our beta cell, we exocytosed it and we said it upregulates GLUTs in lots of other cells. So it makes other cells express more GLUTs, so therefore they take in more glucose and it reduces blood sugar. How it does that is by binding to tyrosine kinase receptors. So tyrosine kinase receptors, you have them in pairs, basically. They're called tyrosine kinase because, remember we talked about amine hormones being derivatives of tyrosine. It's just an amino acid. They're called tyrosine kinase receptors because they have the amino acid tyrosine in residues. And this is on the intracellular end of the receptors. So there you can see we've got two of them, and they've each got their own tyrosine residues. At the moment, they're unbound and they're apart. They've fallen out. They're not talking. Along comes two molecules of insulin, one per receptor. So our insulin binds to each of the receptors. When that happens, the receptors move together. Don't worry about the mechanism, they just do. The receptors move together. And what happens here is they bring the tyrosines from each receptor together to form a dimer. Remember, we use that term in terms of beta and gamma on our G protein. Just means two things together. So these tyrosines now form dimers because we've got one from one receptor, one from another receptor, all lined up. When they form these dimers, it activates the receptor. Bear in mind, we've still got our insulin bound to the receptors. What this causes is self-phosphorylation. It's not the only time this happens. Some channels can self-phosphorylate. There are mechanisms where self-phosphorylation is key. In other words, they suddenly become very attractive to free phosphates floating around the cytosol. So our two receptors coming together, forming these dimers, activates them, and as a result of that, they self-phosphorylate. A load of phosphates rush in and bind to them. Now, this acts a bit like a corner shop. So whereas before our kinases, and note this is tyrosine kinase, whereas before our kinases sent out phosphates to other enzymes, they delivered. They did home delivery. In this case, we're talking about a corner shop. So you have to go and pick up. This is click and collect as opposed to delivery. So the proteins come in and they can collect phosphates from the receptors. When they do that, exactly the same result as all kinases when they phosphorylate, it activates or inhibits those proteins depending on what those proteins are. So that was our protein hormones or our catecholamines, basically our water-soluble hormones that cannot get through the membrane and have to signal via extracellular receptors. Now, lipid-soluble steroid hormones are different because they can get through the plasma membrane. They signal via intracellular receptors. No second messengers required. They can have effects by binding directly to genes. So they can get into the cell and they bind to a receptor that then binds to the DNA. These receptors that they bind to are known as nuclear receptors or NRs. Because once they're bound by their ligand, in this case we're talking about a steroid hormone, they move to the DNA and they bind to something called hormone response elements, HREs, on the DNA. And this activates the transcription of specific sequences of codons. So this will call in RNA polymerase to unzip, to pair the ribonucleotides, and off we go, resulting in protein synthesis. They can also have, it's now known, effects on other areas of the cell. So whereas genomic activity is about activating genes, DNA, this is about non-genomic activity. So this is other actions within the cell. So it's now known that steroid hormones can bind to intracellular receptors that do not run off to the DNA necessarily and have direct genomic effects. So within the cell, they can also signal via kinases or by interacting intracellularly with plasma membrane receptors. So these receptors are inside the cell. And in some cases, actually, this can be an indirect way of having genomic effects. So in other words, what we're saying here is that our lipid-soluble steroid hormones can get into a cell and they can either bind to a receptor, a nuclear receptor, that then binds to DNA, straightforward protein synthesis, or they can activate other pathways within the cell that may result in transcription or may result in other things happening, phosphorylation via kinases, other bits and bobs that happen throughout the cell. So they can have quite diverse effects. In terms of their genomic effects, so this is where we have a nuclear receptor. The steroid hormone diffuses through the plasma membrane, non-facilitated diffusion, remember, so it just diffuses straight through the lipids, no plasma membrane proteins here, thank you very much. It then binds to our nuclear receptor. So this is our receptor here in the big blue blob, and our hormone is the little green blob sticking out of it. So this is it binding to its nuclear receptor. The hormone receptor complex, we call it a complex because we've got two things together, then goes off to bind to the hormone response element, the specific site on DNA that will take that receptor. I suppose you could look at it a bit like a old-fashioned china cup and saucer. We put the tea in the cup, which is our nuclear receptor. The nuclear receptor then fits in a saucer on DNA. This then acts like a transcription factor. And if you remember transcription factors, the next stage is the recruitment of our RNA polymerase. So we end up with transcription of that sequence of DNA. Transcription, of course, leads to translation, it leads to protein synthesis, and that steroid hormone will have stimulated protein synthesis of a protein, whether that be chemical or structural. So our hormone levels then, if you Google hormone levels, you get lots of things about women and menopause. We're not talking about that. Hormone levels are going to depend on the rate of production. So the rate of production and secretion, of course, is going to be dominated by our hypothalamic pituitary control and negative feedback systems. That can control how much hormone we have available going around the circulation. There's also an issue in terms of the circulation, in terms of rate of delivery. So this could be the circulatory rate of the blood, but also the number and affinity of receptors. As we talked about, we can have downregulation, upregulation in terms of both the number of receptors and their attractiveness, their affinity for their ligands. It's also dependent upon the metabolic clearance rate. So the rate of degradation or elimination, how quickly we get rid of that hormone through normal metabolic activity. So we have catabolic degradation by enzymes. So enzymes come along and break down our hormone, either in the cell or in the blood. In terms of elimination, generally we're talking about excretion in the urine or the bile. And the half-life, so the point where we've got sort of half of it left and we've catabolized the rest, the half-life of individual hormones varies. So in terms of adrenal steroids, so this is our corticosteroids, we're looking at around 20 to 100 minutes. So that's quite quick acting. Remember, these are synthesized as needed. They go out, they have an action, we get rid of them. Because they can diffuse everywhere, that's probably appropriate. In terms of our thyroid hormones, they can be bound within the blood and they can circulate around in an inactive form, remember our T4, for quite a long time. Of course, this is also compounded by the fact that we can actually store quite large quantities of these within the thyroid gland. So the half-life of thyroid hormones is anything from 1 to 6 days. So there's quite a wide difference there depending on the hormone. So that's the end of our hormones topic. This is a split week, so you need to start neurophysiology this week as well, before we have our live session. But we are at the end of our third topic, only 4 to go. As always, make sure, please, that you have this one under your belt, you have understood it, you have written your notes, and you have, as much as possible, learnt it before moving on to the next topic of neurophysiology. And it's not accidental that we have hormones and neurophysiology so close together, because of course they are both really key communication systems within the body. I'll see you in neurophysiology.